All right, well, hello everyone. So we're going to begin looking at blood. So what you'll see in the next really several different separate different lectures is we'll be looking at the blood, we'll be looking at the heart, and we'll be looking at the blood vessels, right? And so really all of these together are kind of cumulatively a part of the cardiovascular system. So I want you to think about that and keep that in mind as we go through these various different topics and concepts, right? So just generally as blood as a whole, right? Remember from your previous lectures, AMP1, when you talked about histology, right? Blood itself is defined as a connective tissue, right? So it's defined as a connective tissue because we have different types of cells. Remember, a tissue is comprised of at least two different types of cells working together for some kind of common purpose, right? So we have red blood cells, we have white blood cells, we have platelets, which aren't technically cells, but we'll kind of talk about that. The blood is largely involved in transport, right? That'll kind of be one of the main things that we'll talk about. But we'll talk about it in the role of the immune system. We'll talk about it in terms of coagulation, right? So forming clots of blood, right? So as to prevent massive hemorrhage, right? And then we know that blood is circulated via the heart, and then it moves out through our different vessels, arteries, veins, and capillaries. Just as a little preview for what we'll see when we do our lecture on blood vessels, is you define a vessel based on the direction that the blood travels, not the type of blood in the vessel, right? And so keep that in mind. We won't go into any more detail right now, but we will talk about some additional things later on, right? So within blood, we know we have two main parts, right? We have something called formed elements and we have something called plasma. When you think of formed elements as the things in the blood, primarily the cells, right? So we have red blood cells, we have white blood cells, and we have platelets, like we just mentioned. Now, we'll refer to these, I may refer to them as their kind of general term. So red blood cells, I may also refer to them as their technical name, right? So erythrocytes, right? In this case, erythros meaning red, cyte meaning cell, so these are our red cells, right? White blood cells are leukocytes, leuko meaning white, cyte meaning cell, these are our white cells. And then platelets, you may also see platelets called thrombocytes. And thrombocytes, the prefix thrombo means clot. So a thrombus is a clot. So thrombocytes are clot cells, right? And so we'll look at these in more detail individually later. And then you have the liquid portion of blood, right? The aqueous part, which is referred to as the plasma, right? And so you have your formed elements, you have proteins, you have lots of different things that are dissolved in the plasma, right? So when you hear of, the discussion of like, blood glucose levels or the sodium level in the blood, right? That's the glucose or the sodium or whatever thing you're talking about in the plasma, right? So a normal, sodium level is 135 to 145 milliequivalents per liter or something like that. So you have that much or that concentration of sodium in the blood, but specifically in the plasma of the blood, right? So technically in the fluid part of the blood. So know that these are two different parts of the same thing, right? They're both part of and make up the blood itself, right? Now we mentioned different functions. Again, if you think about these functions now, it will make more sense when we get to specific topics and concepts later. So when we get to blood vessels later, when we get to the immune system later, when we talk about blood clotting later, right? But a big thing is transportation, right? So moving things, moving gases, moving hormones, right? So we talked about hormones in the previous lecture, right? With endocrine system, and we know that endocrine organs and glands are very vascular, right? They have a very rich blood supply because how do you move a hormone from the thyroid or the pituitary to the thyroid? You do it via the blood, right? So we're transporting things, right? Protection, right, with leukocytes and different plasma proteins like antibodies, right? We'll talk about these later on. The platelets that we'll see help protect us in a way, in a sense, right? In the sense that it helps us kind of prevent massive blood loss, right? Regulation is another major role of blood and the things in blood. So body temperature, we'll talk about some of the properties of blood and the properties of water that allow us to maintain a relatively steady internal body temperature. We'll talk about this when we do blood vessels as well, with the size of the blood vessel changing, right? So vasodilation and constriction. the pH of the body, right, and the pH of the blood specifically. We will talk about this, and this is a number. There are certain things in in anatomy and physiology that we try to, not just make you just memorize this, but learn the concept, right? Learn the application. There are other things that you just have to memorize, right? There's not a real good way around it. And this is one of those. So the pH of the blood, okay, commit this to memory, okay? Get it tattooed on you, okay? Not really, but remember this. pH of blood is 7.35 to 7.45. Okay, 7.35 to 7.45, right? We will talk about this more later in the course when we get to respiratory system, when we get to kidneys and things like that, when you talk about maintaining the pH in the body, maintaining the blood pH, 7.35 to 7.45, right? Water balance or fluid balance is another big one, right? So the amount of fluid that we consume, the amount of fluid that we get rid of, right, and we excrete in our urine or our feces or something like that, that's all going to affect or have a influence on our blood volume, which is going to affect blood pressure as well, right? So we'll talk a lot, again, in several separate lectures, but we'll connect them together. Several lectures throughout the course, we'll talk about the role of fluid in the body, right? So let me ask you a question, okay? And you can answer it in the safety of your viewing space. What color is blood? Okay, what color is blood? You can answer out loud, right? What color is blood? Blood is red, okay? Is blood ever blue? Is blood ever purple? Is blood ever any color other than red? The answer is no. Blood is always red, right? Now, the shade of red, as you can see on this image, the shade of red will vary, right? And that depends on the amount of oxygen in the blood, which is a function of the amount of oxygen bound to hemoglobin, right? And we'll talk about hemoglobin in a little bit. But blood is always red. Okay, you may have heard growing up that, when you look at veins or whenever you're talking about blood inside the body, it's blue or it's purple, and then whenever oxygen hits it or whenever air hits it, it suddenly turns red. Well, that doesn't make any sense because if it was when oxygen hits it and it turns red, well, there's oxygen in the blood all the time, right? Even deoxygenated blood, even this blood right here that's really, really dark at the top, there's still some oxygen in that, right? Not as much oxygen as you have in these lower blood samples, but there's still some oxygen in it, right? So that explanation really doesn't make any sense, right? So blood is always red, okay? The amount of blood is also going to vary depending on the size of the individual, right? So normally maybe somewhere around 5 liters, but obviously it's going to depend on your size. It can typically vary based on sex as well. So males on average tend to be larger. They tend to have more blood. Females on average tend to be a little bit smaller. They tend to have less blood, but there's obviously some variation. There's lots of variation in that. And then blood also has what's called a viscosity. Right. So essentially, when you think of viscosity, the synonym that you can think of is thickness. Okay, thickness. Technically, viscosity is a fluid's opposition to movement. Okay, so to what extent does this fluid oppose moving? Right? Does it oppose being moved? Right? Does it oppose being moved through the heart or through the blood vessels or something like that? Right? And so that's going to be a function of the amount of things in the blood. Right? The amount of cells that we talked about earlier, right, which are part of our formed elements. The amount of proteins in the blood, the amount of solutes and whatnot, right? It's going to vary, right? So a big thing is the number of erythrocytes, so the amount of cells, and the amount of fluid, right? So think of this as if you added a bunch of, so say you have a bowl of honey, okay, or some other thick fluid, maybe syrup, and then you added a bunch of water to it. Right? You add just water. You have a bowl of syrup or honey, and then you dump two or three cups of water. Right? Is that solution, right? Is that honey or syrup? Is it going to be more thick or less thick? Right? It's going to be less thick. Right? So it's going to, in other words, you could think of the viscosity as decreasing as you add an additional amount of fluid. Right? So the opposite is true is if you take fluid away. Right? So the viscosity would increase if the amount of fluid decreases. Right? Example of this in our body, this is dehydration. Right? If you're dehydrated, the viscosity of the blood is going to increase. And then likewise, the viscosity will increase if the erythrocyte number increases. So in other words, you begin making more red blood cells, and we'll talk about some kind of clinical application of this in just a little bit. Right? Some characteristics of blood, right? We already mentioned this earlier, that there's a certain concentration of things in the blood, so proteins and ions and other molecules like glucose, things like that. And those concentrations are obviously going to change based on changing the amount of that molecule or that ion or that protein itself, right? So if you eat something very sugary or have a very sugary meal, right, or a carbohydrate rich meal, then you're probably going to increase the blood glucose level, right? Likewise, if you alter the amount of fluid, right, and keep everything else the same, you're probably going to decrease some things as well, right? And so we'll talk about how that influences the movement of water into and out of the blood vessel and into out of the cell as well. Blood temperature as well. So blood is approximately 1 degree Celsius higher than measured body temperature. It's warm deep inside the body. And so we see this in a more relevant discussion in terms of blood vessels maybe dilating to bringing more blood closer to the surface to allow for the dissipation of more heat, right? So to help with thermoregulation, right? And then as we mentioned earlier, blood pH, again, that 7.35, 7.45 is critically important, and we'll come back to that several times during the course, right? When we look at blood, you can take blood. So if you took blood out of your body right now, right? So the images that we looked at earlier, if you ever go into a clinical setting or go to your doctor and they take your blood, right? They take a blood sample. That's whole blood, right? So meaning your whole blood, right? Meaning everything, right? We're not separating anything out yet, right? It's everything in your blood. It contains red blood cells, it contains white blood cells, it contains platelets, it contains proteins, it contains everything, right? You can take that whole blood and you can spin it, okay, in a machine called a centrifuge, and by spinning it, you're separating out the different components of the blood based on relative density, right? So you get erythrocytes at the bottom. Why do you think erythrocytes are at the bottom? Okay, think for what's some reason that something may settle toward the bottom of a solution, right? I'm not going to answer that, but you think about that. What is something that could cause the red blood cell to settle toward the bottom, right? And this is a good chunk of the overall blood sample, 44, 45%. You have a middle layer that's very, very small called the buffy coat, and this is made of leukocytes and platelets. It appears typically kind of clear sometimes, or kind of yellow. And then you have the plasma layer that settles at the top. Okay. And again, for the same reason that the erythrocytes settle at the bottom, the opposite reason is why plasma goes to the top, right? So here you can see what you may refer to as a centrifuged blood sample, right? So again, in the vial, you take whole blood, you spin it in the centrifuge here, and you separate out, again, based on relative density, the plasma, the buffy coat, and the red blood cells, right? And the red blood cells. So you can see what you would expect to find in each layer. So plasma, about 55%, okay, of whole blood by volume. Red blood cells about 44%, and then that remaining 1% or so are your white blood cells and your platelets. Okay. Now, if you take that centrifuged blood sample, right? So if you take this value, you can then describe something called the hematocrit, right? And the hematocrit is essentially the percentage of the volume of blood that is made up of formed elements, right? So remember, what are formed elements? Formed elements are, remember, like the things in the blood. Specifically, think of the cells, right? So think red blood cells, think white blood cells, think platelets, right? So percentage of volume of all formed elements that makes up blood, right? Well, what did we just say? If we look at our components of our centrifuged blood sample, we've got some white blood cells here, we've got some platelets, and we've got some red blood cells, right? So this is making up about 1%, and this is making up about 44%. So we could say together with our white blood cells, our platelets, and our red blood cells, that's somewhere around 45%, right? And so it's going to vary somewhere in there, right? Males, 42 to 56%, females, 38 to 46%. It's somewhere in that given range, right? Now, you'll notice here that I have a bullet, clinical hematocrit. Right. So clinical hematocrit is a more clinically relevant uh, version of the hematocrit, if you will. And essentially what that is it's saying what percentage of the volume of whole blood is made up of specifically red blood cells, right? Well, if we go back and we talk about formed elements, okay, so our formed elements, again, erythrocytes, white blood cells, platelets, red blood cells are making up the vast majority of all formed elements, 98, 99%, something like that, right? So essentially, when you're talking about hematocrit, and you're saying the volume, percentage of volume of all formed elements, you're really saying what percentage of the overall blood is made up of red blood cells, right? Made up of red blood cells. Okay, that's the clinical hematocrit if you hear that, right? A blood smear is when you take a sample of whole blood and you smear it on a microscope slide. You can look at it under a microscope, and again, you can see various types of cells in that blood smear, right? So here we see a blood smear. We can see all these red cells here. These are erythrocytes. These are red blood cells. We can see various types of white blood cells. We'll look at the various types, and then these very small fragments of cells. That is, or those are platelets. Those are platelets, right? This graph to the left here, what you can see here is this is looking at viscosity as a function of hematocrit, right? So in other words, as you increase the hematocrit, right, which again, think to yourself, what is hematocrit? It's the percentage of whole blood that is made up of formed elements, which is essentially like saying the percentage of whole blood made up of red blood cells. As you increase that, so in other words, as you have more and more cells in the blood, you're increasing the viscosity, right? So we're increasing hematocrit and we're increasing viscosity, right? So we have this kind of gradual increase in the thickness of the blood, if you will, as we increase hematocrit. So we don't want hematocrit to be too high, right? We can actually have too many red blood cells, but we also don't want it to be too low because then we're compromising our oxygen carrying capabilities, right? So there's a happy medium, and again, somewhere in these different ranges here, depending on age, depending on size, depending on sex as well, right? So there's various things to consider, right? So we're going to stop here, consider some of these questions, and then we will pick up with our next part, continuing with this lecture.